All right. Well, good afternoon, everyone, and welcome to today's Exploring by the Seat of Your Pants event. My name is Joe Grabowski, and I'll be your host for today. For those of you who are new to our program, we bring those on the front lines of science, exploration, conservation, and adventure live into classrooms around the world. So today we have a really special treat in store. We are heading live into the field to connect with the National Geographic Pristine Seas team as they partner up with the Waite Institute and Nurajeh to research the remote southern islands and atolls of the Maldives. We're going to head on board the research vessel Blue Force 3 and meet some of the team that includes scientists and filmmakers, both local and from around the world. Kicking us off, starting things for us today will be Kieran Fraser. He is the expedition leader. Let's bring him in live right now. Hey, Kieran, how are we doing today?
Hi there, Joe. Yeah, absolutely fantastic. So we're sat on the expedition ship Blue Force 3 with colleagues from Pristine Seas and Nurajeh. We're sat in Kukumbula Harbour. We've been working in Hoodoo for about 6 or 7 days, and after tonight we're heading on down to Adu Atoll. We've been at sea so far for about 12 days, and I think the total expedition length is going to be about 21 days. We're really excited to be working with our partners in the Maldives to help better understand the oceans around the Maldives and help the government protect them. So if I hand over, what we're going to do is work around the ship, meeting various scientists over the next few minutes. So if I hand over to our chief scientist, Alan.
Hey everybody, good afternoon. I'm Alan. I am the chief scientist on this expedition. I'm from Hawaii. And chief scientist on the expedition, think about a conductor in an orchestra, making sure that all the instruments are playing correctly. And so we're doing a lot of different fun science here that's really going to be important and valuable to the Maldives to help manage the beautiful coral reefs that you have here. So we're doing everything from scuba surveys to remote camera systems. We're filtering for eDNA. And what we're going to do is take you around the ship and look at all the different activities that we're doing on board the vessel. So for me particularly, I'm working on fishes. So I go underwater and I count fish. There's a lot of fish in the Maldives. We've counted over 300 species so far, so it's really rich. It's exciting to see all these new and different species. The Pristine Seas team has been going for 15 years. We have gone everywhere from the North Pole to the South Pole and every place in between. We've had over 40 expeditions to some of the last wild places in the world. It's our first time in the Maldives. We're really excited to be here. And now I'm going to turn it over to my colleague, Maisha, who's with Nurajeh, and she's been leading the coral work that's been going on here. So Maisha, why don't you tell them what's been going on so far?
Thanks, Alan. Hi, everyone. Thanks for joining in. I'm Maisha. I'm from Nurajeh. Nurajeh is a partnership between the government of Maldives and the Blue Prosperity Coalition, and the Pristine Seas is partner to the Blue Prosperity Coalition. Like Alan and Karen said, we're here in the southern parts of the Maldives, currently in Fomalhaut Harbour, and we'll be heading to Adu Atoll later today. And we have been doing surveys in Hoodoo and Fomalhaut atolls, and I in particular have been looking at the benthic composition, the surveying the corals, and we have had some exciting dives. I'm really excited to show you around the ship, what else we have been doing here with the team.
Great, that's awesome. Now, so Maisha's talking about, she's been working on corals and helping to lead the expedition. And now I'd like to introduce our colleague, Quique Ballesteros from Spain, who is working on algae. So he's the algae specialist on the trip. And Quique's got some fun things to show you.
Hello, how are you? My name is Quique. I come from Spain, and I want to show you some algae. And they are very important for the reef, mainly because they produce structure of the reef and also oxygen. You know that oxygen is very important for all the life on the planet. So without oxygen, we couldn't live here. And we will begin by these algae. Okay, these are algae, green algae, that they are calcareous ones. and they are the main organism that make the sand of the beaches from Maldives. Not the corals that make rubble, but the Alimeda talus, when they die, they stay white like this, close up, and they decompose and they make the sand of the beaches. Another one that I like especially is that one. Look at that one. You see? This is not calcareous, so it doesn't serve almost for nothing, but probably there is some fish that feed on them, because we have seen some fishes around and probably they are feeding on it. And the scientific name of this species is the algae of the expeditions. So I think it's very nice to have an algae that is named after the scientific expeditions. Okay? And finally, I want to introduce you to that. That you would say more, or we would say this is a rock, and it's hard. It's very hard. Okay, and it's not a rock. It's an alga, a seaweed, but it's made by calcium carbonate, and it has the same role of corals. So they build the reef and help the corals to build the reef. And what color is that? It's red. So you have red algae, mainly in the coral reefs, and green algae. Different colors, different groups of algae. That's it.
Yeah, excellent. Thanks a lot for that, Kike and Alan. We're going to go outside and speak to one of our scientists, Whitney, now, who's going to talk about drop cameras.
Hello, everyone. I'm Whitney, and I'm going to show you some of the cameras that we use for science on our expeditions. So these cameras here, these are special cameras. They're not the ones that you would throw in your backpack and take photos of your family. They are deep sea cameras. So they're about the size of a beach ball. One person can carry it. And these cameras, what makes them special is that they can go down to, they can go super deep. They can withstand the pressure at the bottom of the ocean. So they can go down to the very bottom of the ocean, over 3 miles deep. And they film animals. So you can see right there, there's the lens of the camera. So that films animals on the deep sea floor. So the way it works is these cameras, we attach bait to it. So we'll put dead fish in a can, and that's going to attract all the animals to come to it. So the cameras sit like this, if you can get the bottom there. So the bait gets attached here. So all the animals are coming to the bait, they're all attracted to it, and then the camera is recording. So any animals that come to see that come to try to get the bait, they're on camera. And so after the mission, we leave these down on the bottom of the ocean for about 5 hours recording, and then after the mission, these glass balls come back up to the surface. And once they're at the surface, then we pick them up out of the ocean. We download all of the footage, and then from our own computers, we get to see what's on the bottom of the ocean. Oh yeah. And so as they're sitting on the bottom, because the deep sea, there's no light down there. The light doesn't reach, so it's super, super, super dark. So in order to film the animals, we need to have lights. And so these cameras are designed with these arms that come out. Oops. So these arms, when they're at the bottom of the ocean, they shine light, and they're really, really bright. If I were to turn them on right now, it would blind all of us. They're so bright, but they can light up just enough so that we can see what animals are down there.
That's really cool, Whitney. How many times have you deployed them on this trip so far?
In the Maldives, we have 8 deployments so far. So 8 different days we have gone down to see what's down deep.
What's the deepest depth you've gone?
So far, the deepest we've gone is 1,800 meters, which is that like 2 miles?
Just over a mile.
A little over a mile. Yep. There's no light down there, and the animals that are down there are completely different than you would see up here at the surface.
Cool. Thanks, Whitney. That was awesome, everybody. We'll get some questions in a minute, but now I'm going to turn it over to Chris and Juan. These are the guys who have been deploying these cameras in both on the surface of the water and also at shallower depths than the one Whitney's doing, but they're doing similar things. So take it away, guys.
Yeah, so basically Alan and Jen and the Bentley Team scuba divers have it kind of covered in the inshore, and Whitney has it covered in the deep sea, but these cameras let us look at things that are out offshore and a little bit deeper than the scuba divers are going. So basically what we do is they have 2 GoPros and they're separated out apart, and they've got an angle on them, so it allows you to have stereo vision. So basically like your eyes can tell distance and size of things. It means that with these cameras we can tell the size of the fish, how many there are, and what sort there are. So these ones basically will go down to the seafloor, and they have cameras and lights that can go deep. And then these ones we deploy, which are the same, but they go offshore. So we go out into blue water. So if you were to go out there scuba diving, you'd be waiting a long time until you sort of saw a fish, and it'd take a long time. So we go out there with these cameras, and they also have bait, and we leave them for a couple hours, and that lets us see what might be getting around offshore. So that might be tuna, might be steelfish, it might be sharks or dolphins or whales. So we see all sorts of different things offshore, and nice tool to allow us to do that.
And when we throw these a little bit deeper, between 30 and 60 meters, where we still get some sunlight, it's not pitch black like the ones where Whitney works, we get to see different types of ecosystems. For instance, here in the Maldives, at about 40 meters, we saw a lot of soft coral along a very impressive vertical wall. And that is something that we have not been seeing in the shallows by our divers. So these cameras, when deployed between 30 and 60 or 70 meters, allow us to fill in the gaps in between what the divers are seeing and what we see in the infrared drop cams.
Cool. Thanks, guys. So now we're going to go. So Juan and Chris and Whitney have been talking about the deep sea. We've been talking about the shallow scuba dives. Juan's been working in an area called the twilight zone. So that's where there's just a little bit of life left, and the ecosystem's really different. Chris is working on the surface where we're seeing big animals. Now we're going to go up and see things that we can't even see at all with the naked eye. We're going to go up and look at what Molly's doing with water to talk about some eDNA, environmental DNA. Let's go upstairs. Okay, take it away, Molly.
Hello and welcome to the filtering station. So I look at the tiniest little stuff, the cells, the DNA. And you know, if you clap your hands, there are cells that are spreading all over the place. Similarly, in the ocean, fish are pooping, and there's mucus, and there's plankton, and there's shedding of scales. There's all sorts of DNA just floating in the water column, but you can't see it. However, we can take water from that water column, and we can filter it out and capture that DNA on a filter. And actually, it's right here. This is the filter that we use. And so by capturing it on the filter, we can then extract, meaning we can pull all the DNA out of the cells and look at the code, the sequencing code, and then we look at it to see what's there. Because each organism has its own DNA fingerprint. Kind of like you and I have our own fingerprints that determine who we are. It's the same thing with DNA, so we can look at different species. And here we've got these pump systems. And so what's happening, these filters are attached to tubes. They're inside the water. The water is being pulled out of the tube, going through this pump, and then it's pouring out on the other end. Do you see the little tubes pouring out here? And this is all the excess water. So essentially what we end up with are all of these samples that have different DNA from different biological organisms from the water in the Maldives.
Awesome. Thanks, Molly. So let's head back downstairs again, and we're going to see what some of our other colleagues are up to. Hold on. Come with us. Okay, guys, you're on.
Hi, everyone. I'm Katie. This is Nicole. We're from the Scripps Institution of Oceanography, and we're going to talk to you about photo mosaics. So if you want to check out this video, this is a video of what we've been doing underwater. Sped up, we don't actually swim that fast. But essentially, we swim around with a camera rig. You can see this person has 2 cameras in their hands, and we swim back and forth over the same area, collecting thousands of images. And then we use a computer program to stitch all of those images together to make a 3D model of the reef. So that's sort of a way that we can kind of pick up a chunk of reef and bring it home with us. And then we can ask all sorts of questions about that little section of reef. So Nicole has an example of one of our cameras. So we'll swim around with 2 of these that are taking pictures at different amounts of zoom on the reef, so we can get some really detailed pictures and then some wider angle pictures. And they'll all get stitched together into a model. And here's an example of one of the models that we made last time we were here in the Maldives. So this is a model from Fumala. We actually dove there today and found the same pod. So I'm going to change to a different video and hand it over to Nicole to tell you a little bit about the science.
Yeah, so a couple of years ago, we were able to come here to the Maldives and collect some of these images. And so part of the thing that we're doing on this trip is not just collect new images, but also revisit some of these places that we went to a couple of years ago. So you can see on this video behind me, this is what we hope to learn from all of these images. You can see these corals and you see these blue halos around them, which is showing the growth. So by being able to come back and image these same exact areas, we can track how much these reefs are changing, whether in a positive way with these blue halos of corals growing, or maybe there's a storm event with these red splotches where some of the structure in the reef, some of these corals have been dislodged. And so we can track all those changes, good and bad. Both of it is happening here in the Maldives. You can see a lot of blue growth around these corals. There's a lot of growth that's happening, and all of this is happening in just a single year. So we can track a lot of these big dynamic changes of the reef in just these short time periods with these images.
Hi, my name is Hussein. I'm one of the local guys who is participating in this expedition. I think the work these guys are doing through the Blue Prosperity Coalition, NORAD, and all other partners of the project are doing very important work, because we talk about conservation and protection. I mean, unless we know what we are conserving or protecting, we will not have any idea of, I mean, the importance of protection. So, for example, coral reefs we have in the Maldives, we have over 2,000 square kilometers of coral reefs from various studies and so on and so forth. So it's a large area, and the local community or local expertise doesn't have the capacity to do this kind of work. So this kind of expertise, scientific knowledge, and everything that comes as part of this exercise becomes very important for the Maldives to do the marine spatial planning and also eventually to the protection of the reefs. I will say a little bit in Diwali as well, because I think it's important for this, because I mean, NORAD program ki Divaraj me ane Nunia blue economy Nunia blue Maldives egota tarjuma kare in the program hai. Me program ki astuga me deruga me මෙකමක තජරබාවර බේෆුල්නා ගුලෙන් දිව රාජ්‍යයක marine special player इन्होंने हासप राज्य में ना दूसरे में जो ईज़ेडा हमारे दिव राज्य कानों के बेड़ों में हेवगत का प्रविधान में वता का मालूमात एक को हज़ारा मुसल्ली करो मसक्त तेरे वारन दीना मालूमात तक में निकुमे के अन्ना ने स्कूल को दिन ना बस वारा मुहिम काम में निकमु का शाबुवरी काम मेरे तेरे में अच्छा करने को में मिलो ना हम पुरम्बेडु वाले पुरम्बे असको बैवर मसक्के न बोकरे देगा में वारा पुरपुर को एक याद मिसाल का EDNA कोटे तो इंगेटी दुनिया में कैमरा तक हम कोटे तो है पूरी में स्कूल है मिसाल का दूसरे वारा कंप्यूटर आडियो दिया हम एक को तक में दुनिया में इरुगा अलिका में ऐसा को उड़े धीरे अच्छे में बहुत नहीं असलो अली बेड़ों को भी कैमरा बेड़ों को भी इंसान में ड्राइव कर रहे हिसाब है उसमें अड़ीगर में होना बाइक जिताना में आसमान में से अनुनिया सुपास होती है मालूमात है तुम के बारे में बताओ।
Thanks, Hussein. Great, thanks a lot to Hussein and the rest of the science team. Joe, have you got some questions for us that you'd like answered?
All right. Well, let's change gears and let's jump into a little bit of Q&A action. I want to give a shout out to everybody who's tuning in live right now. I can see we've got a good group tuning in live. I know a lot of the groups are local from the Maldives, so don't be shy to use the chat. It's a live chat, so introduce yourselves. Let us know where you're tuning in from. And then, of course, send us in some of your questions because we want to see those questions, hear what's on your mind, and the whole team is standing by. So, Kieran, it is an amazing tour that we just had. There is so much science. You have such a great team there with you. I'm wondering, what is kind of a typical day for you as the expedition leader?
Thanks, Joe. Yeah, well, a lot of us are up quite early, so typically I get up at about 6 o'clock, 6:15, and then usually Chris and Juan, who you saw just now, and Whitney are usually away in the first couple of boats to go and put their cameras in the water by 6:30. Then usually around about the same time we're having breakfast, and then the dive teams, who includes Maisha, Hussein, Alan, and many other people on the vessel, are usually heading off diving on our large and very nice Donny at 7:45. Everybody tends to finish a lot of that work by lunchtime, which is usually about 12:30, and then the afternoon we're usually running science dives again. And also alongside, who you haven't met today, we've also got a media team who are producing a short film to be shown in the Maldives. So we have quite a long day, and then usually eat in the evenings at around about 7 o'clock, and then after that I give a quick briefing on what's going on the following day. So every day is pretty busy. We work every single day during the week, 7 days a week.
All right. So let's start taking some of the questions here that are starting to come in. So the first question I have here is about the weather. How's the weather been on the expedition so far?
I can probably deal with that one. Yeah, I mean, the weather's been amazing. It's been pretty consistent. We've had mostly winds from sort of northeast, north through the east, and it's been really calm. We haven't lost any days due to weather, which is really, really unusual on this sort of expeditions. And for somebody coming from Devon in the UK, at the moment there's snow on the ground, I think. So to be out here in sort of 30-odd degrees is really, really nice.
All right. Okay, so another question here about the overall health of the coral. How is it looking so far on the reefs you've been exploring?
The reefs of southern atolls have been, we noticed it's very, very diverse. We have actually had a very exciting dive where we found a colony of very rare species of coral, which is thought to be locally extinct since 1998 bleaching. We have a few sightings from different parts of the Maldives, but seeing them here in the southern atolls is very, very exciting. Hussein, do you have something to add?
Yeah, I think it's important for the students to know that we had a very devastating coral bleaching event in 1998. And since then, the reef have been recovering quite alarmingly in some atolls, but more importantly, the southern atolls, the recovery is like 6 times higher than the northern atolls. Like if you compare to the north of Maldives to Adu Atoll and Hodu Atoll. This is the first time this kind of expedition being done in or survey being done Formula. But Formula being a very exposed area, the coral community is somewhat different from other sheltered areas where the reefs have more sheltered reef areas. But as Maisha mentioned, some of the corals, especially the fine branching corals, actually almost vanished from the Maldives. And then I have been actually challenging people if you find Stylopora or Histiophora, then I will give a reward for people. But fortunately, since then, these guys have found this Stylopora islata from Hodu Atoll, I think.
Hodu Atoll, and I think it was on the previous expedition in 2021. We had an expedition here in the southern atolls, and Scripps actually had one of their sites where you guys wanna talk about the site where we found the Stylopora?
Sure. Yeah, one of our mosaic plots actually had a colony of Stylophora right in the middle of it. So we had been looking really hard while we were diving in 2021, and it happened to fall right in the middle of our plot. So that's really cool. We went back and resurveyed it again this year, so we'll be able to track that colony over time and see if it grew, how much it grew, and how its health is doing. So that's pretty cool.
So with those mosaic plots, how big of an area are you looking at each time?
Yeah, the mosaic plots are 10 meters by 10 meters. So about the size of like half of the tennis court or basketball court. Maybe an area about the size of this room that we're sitting in here on the ship. So it's a pretty decent area. We're hoping to capture anywhere from hundreds to thousands of colonies within that plot, depending on how healthy the reef is and how many colonies you have in there. And one other thing that we've been able to find from the mosaic plots that has been pretty cool is we track coral recruits, so baby corals. And when we were here in 2021, we found that there were a ton of baby corals down in the southern atolls. And we went back to some of those sites this year, and we saw not only more baby corals, but actually a lot of sort of teenage corals as well. So we're hoping to track those and see if those are some of the baby corals that we saw last time.
All right. Well, we have a couple shark questions here on deck. Have you come across any sharks yet on this expedition?
Maybe the Brooks.
Yeah, we've seen a few sharks on the cameras. So we've had, on my cameras, I think we've seen silky sharks, tiger sharks, and we had one short fin mako as well. So that was really nice to see. Mostly down around Fulmula here, it seems the busiest. And I think in the deep sea we've had some as well.
Yeah, we've also seen some deep sea sharks. So on a couple of the deployments with those big deep sea cameras, we've seen 6-gill sharks, which are sharks that they don't come up to the surface. They are deep sea. They look really different from sharks that we would see up here. We also saw a lantern shark, which is also another deep sea shark. These are animals that are very different from what's at the surface. And the lantern sharks, they're not that big. They're maybe like this big, and they're called that because they have these things on their head that kind of glow. In the dark, down in the deep sea, when our lights shine on them, you can see these glowing bits on their head. But yeah, the sharks that we see in the deep are very different from up here in the shallows.
And then in the twilight zone, in the reefs in the twilight zone, we have seen 2 species of sharks that are commonly seen by divers too. These are the gray reef shark and the white tip reef sharks.
All right. So the follow-up question here is, why are those sharks important for those reefs?
So, I mean, sharks are part of all ecosystems, and so you need to have predators and prey and have the right balance of all those different organisms in the ecosystem so that it remains healthy, right? So you need, if you have an absence of predators, you could have an explosion of prey species, and then the system gets out of balance. So when all the components of the ecosystem are there and present, the system can rebound better from a disturbance event like a storm or anything else like that. But just it makes the system operate more effectively and more efficiently. The predators eat the prey tend to grow faster, and don't live as long. So it just to have a healthy ecosystem, you need all the pieces of the puzzle to be in place.
All right. So I know you're roughly halfway through the expedition. You mentioned finding that species of coral that was believed to be locally extinct. Are there any other highlights that have stood out so far?
I think, like Hussein had mentioned, because the reefs were devastated in the 1998 bleaching event, to see the recovery and to see what the Scripps folks have seen just in the short period of time, we don't realize how quickly these things can rebound if given the right chance. So I think, to me, the most exciting thing has been how vibrant the reefs look and how healthy everything looks, especially compared to what happened previously, because that event was pretty devastating.
All right. This question here is from a grade 9 student, and they're wondering, what do you do for fun or free time on the ship? Do you have any?
Enter data. Yeah, it's, as Kieran mentioned, it's pretty much start to finish. We wake up early. The teams are away really early. We're diving. We put several hundred hours in underwater already. So we try to talk about what we see during the day, in the evening, but it's pretty much nonstop till we go to bed. Eat, sleep, repeat, and dive and deploy cameras. But
all right.
It's all fun, so that's what we're doing for fun.
And this afternoon, Joe, we are, because we've been diving for quite a few days now, we're going to take the afternoon off and are lucky enough to be having a look around Formella to actually see some of the islands. Really, most of us, apart from being in Mali at the start of the expedition, haven't got ashore yet. We've not had the time. So we're really looking forward to meeting some of the local people and having a look at some of the local wildlife and sites.
Okay. So we saw a lot of really cool gear. We saw the eDNA equipment. We saw the drop cams. Does that gear always cooperate? Are you dealing with malfunctions? Do you ever have trouble retrieving the drop cams? What's going on?
We got a great answer for Whitney.
Well, working in the ocean is extremely difficult. And when you mix electronics and the ocean, that's even harder. So we kind of expect that there's always going to be challenges with when you're dealing with cameras and technology in the ocean. There's always hiccups, and it's just kind of a nonstop exercise of problem solving and patience. But it also, if it were easy, everybody would be doing this. So it's also really satisfying to kind of meet these challenges and find ways around them, find ways to continue to succeed. Also, with some of the equipment that we're putting out in the ocean, we're putting these things out that are relatively small. The ocean is huge, and when you have like chop and swell and storms, and when you have different ocean conditions, it's really hard to see your equipment in the ocean. So it's also tricky finding them sometimes. We have different challenges with finding our equipment in the ocean too, but we find ways around it.
Excellent, thanks guys. Any more questions, Joe?
Yeah, absolutely. The question is about the food on the ship. How's the food on the ship, or do you have to resort to eating some of Kiki's seaweed?
No, we're really lucky. I've spent a lot of time on ships over the years, and the food on this ship is absolutely excellent. So I think the biggest problem is for all of us not to eat too much while we were aboard. But yeah, the food's great.
All right, perfect. Let's swing back to the eDNA.
the normal food. So green salad, white salad, some carrots. What I am eating usually.
A little bit of salad dressing, that's probably pretty good.
Very healthy.
Absolutely.
Right from the ocean.
Looking back at the eDNA, we saw all of those samples. How long is it going to take to get through and to process those samples?
It takes about 30 minutes for all the filtering to go through one of those small little Sterovexes. But once it's filtered, it doesn't mean you're done. So we have to put something inside those filters to preserve the DNA. And then we have to seal it up and we have to transport it back. And then you're at a lab. And in the lab, it's going to take several weeks to go from extraction to getting those sequences. So the whole process takes a couple months.
All right. And do you find that you can generally identify what you're seeing, or are there times where it comes back as inconclusive or, you know, an unrecognized species?
It's kind of a mix. So one of the limitations with this method is that we need to get what's called a barcode of all the species to really identify it. So like here, you know, if everyone gave their fingerprint, we would be able to identify it. But if half the people here didn't provide their fingerprint, we would only know half of the people in the room. So it's the same thing with DNA, is that we need to go and individually collect organisms to get their fingerprints to put in what's called a library so that we can compare our sequences. So when we don't have an identification, we can still make a species approximation. So we have a sequence, we don't know the species, but we know it's, let's say, a sponge, or we know it's a certain type of fish family. We can at least identify it to that degree. And then the beautiful thing actually about DNA is that in 10, 20 years, we can take these sequences and reanalyze everything once there's more information of these individual fingerprints.
All right. So I've got a couple more questions here, and then I'll let you guys get to your afternoon off. The first one is about human impacts. Are you seeing any evidence, maybe plastics, maybe fishing gear, things like that?
Alan, I don't know if you'd like to comment on that one.
That's a tough one. There's a lot of people in the Maldives, a lot of uses of the ocean. People, you know, are eating from the sea. There's tourism, dive tourism, other types of tourism. So there's going to be human impacts, and those impacts are going to be obviously greater where there's more people. You see greater impacts around Mali than you do in remote places, especially some of the remote islands that we went to in Huvadu. So we see, we see signs of anchor damage. We see plastics in the ocean. Those aren't necessarily locally derived. They could come from, hundreds, if not thousands of kilometers away. And there's, fishing impacts as well. And so it's always to find that right balance between human use and ecosystem health. And those are just things that marine spatial planning will help. Everybody needs to use the ocean wisely. And so it's a tricky question, especially as the global population and the Maldivian population increases over time. So it's a tricky one to answer. We definitely see signs of human impacts, but we also see places that are extremely healthy. And so having that balance and having the ecosystem still in a moderately stable state is a good thing.
Yeah. All right. So you're out there, you're collecting all kinds of great footage, great eDNA material, using the drop cams and things like that. When the expedition ends, the work's not over. So what will be happening when the expedition wraps?
So so we'll be heading back to Mali at the end, and all the scientists will be working on their data. And we're hoping to have an event at the end of the expedition with people in Mali. And we should be showing a short film on what we've been doing on the expedition to give people a bit of a taste of what we've been doing. And then obviously the data that we're collecting in the Maldives is really Maldivian data, so that the information we're collecting will be shared with all of our partners in the Maldives, which is really, really important.
All right. Well, before we wrap up for today, and this is kind of a little selfish on my side as well, because I'm here in Canada, and it is very white and snowy and cold. So I think we should go back on the deck for just a moment and maybe just get a look at the harbor, soak in some green, soak in some sun for a minute before we say goodbye to the team.
Lovely. And hopefully, if it's all right, Joe, we'll just get Hussein to say a few words as somebody who's worked in the Maldives for many, many years as we go out onto deck.
Perfect.
Show us around? Yeah.
Can we go up? Yeah.
Upstairs?
Yeah.
Show the waves.
Tour guide?
Yeah, go upstairs.
All right, I just saw a whole pile of gear. Can you give us an estimate? How much gear do you have jammed on that boat? How much do you think that all that gear weighs?
An awful lot. How much is the gear work, Doug? You can tell us. Our cameraman can tell us.
Probably about 5,000 pounds of gear came from the United States for this expedition.
Wow.
So,
There we go. Beautiful.
So we are on the upper deck of the
Blue Force 3.
Blue Force 3, the expedition team. We hope that you guys got a quick snapshot of what all types of activities we are doing. So it's been great to provide you with this information, and I hope you guys have a little bit of learning of what people like Christians are doing to understand the environment, especially the marine environment of Maldives. Yeah. Thank you.
Well, listen, thank you so much for introducing us to your country, the amazing ecosystems, the undersea world. I think there's probably a lot, well, myself for sure, very jealous. I know there's probably a lot of students who are tuning in today who are excited to get out and explore the world around them. So we had about a dozen groups today, students ranging from 12 to 16 years old tuning in, which is always so great. A huge shout out to the team. Thank you so much for the passion that you share for the work that you do, showing us all the amazing gear, taking us into your life on the ship. It was an absolute pleasure to connect with everybody out in the Maldives today, and best of luck with the rest of the expedition. I can't wait to hear some more stories.
Great, Joe. Thank you very much. Really great to speak to everybody.
Have a great day.
All right. Thanks so much, everyone. We're going to sign off for today. Thank you.
Bye.